Have you ever wondered how it's possible that you can see, taste, or hear? I mean, really, how do these things work? These are all examples of special senses. Special senses are different from the general senses, like touch and pain, in that they use specialized organs, all of which happen to be located in our heads. In this chapter, we'll explore how our special senses actually work. Welcome to part 1 of the voiceover lecture for Chapter 8, The Special Senses. In this chapter, we'll cover taste or gustation in Section 1, olfaction or smell in Section 2, hearing in Section 3. In Section 4, we'll talk about equilibrium. Equilibrium is the sensation of how our head is oriented and whether or not we are accelerating. In other words, equilibrium gives us a sense of balance. Finally, we'll discuss vision in Section 5. Section 1, gustation or taste. Gustation is the sense of taste. It's a chemical sense in that we are actually detecting molecules and ions in the environment. And we only actually have 5 different types of taste. Salty, which detects sodium ions. Sour, detects hydrogen ions. Remember, hydrogen ions are released by acidic substances. So the detection of sour is really detecting acidic molecules. Sweet, which detects sugars, like the monosaccharides and disaccharides. Bitter, which detects several different substances, including some poisons, and some molecules that are derived from plants, like caffeine tastes better. And then finally, umami. You might not have heard of this name, but umami gives foods a meaty or savory flavor. It's detecting certain amino acids. Umami is kind of a beefy or soy sauce sort of flavor. And now for the anatomy of gustation. The receptors for taste are found in structures called taste buds. We have thousands of taste buds. They're found mostly on our tongue, but also occur on the soft palate, that's the soft portion of the roof of your mouth, the pharynx or throat, and the epiglottis, the flap that closes over your airways when you swallow, so food doesn't get down your trachea. The number of taste buds, unfortunately, declines with age. Taste buds are found on structures called lingual papillae. What are lingual papillae? They are the elevations that are found on our tongue, and they give it a rough appearance. In adults, there are 3 different types of papillae, or papilla. The filiform papilla or papillae are found all over our tongue and do not contain taste buds. Fungiform papillae are found mainly near the tips and edges of our tongue. As the name implies, they are shaped sort of like a mushroom, and they do contain taste buds. And at the back of our tongue, in a V-shaped row, are the valate papilla, and those also contain taste buds. Here's a cross-section of the surface of the tongue, demonstrating all 3 types of papilla. The ridge-like filiform papilla are found again all over our tongue, and their function is mainly to increase friction so that we can move food around in our mouth. The fungiform or mushroom-shaped papilla, and the larger dome-shaped valate papilla. These little blue circles are the taste buds. Our tongues are covered in taste buds. Taste buds are oval-shaped structures that are composed of several cell types. You can see in the diagram to the right a taste bud composed of several different types of cells, but we're going to focus on one main cell type, the gustatory receptor cell, also known as a taste cell. Taste cells have tiny little structures called gustatory microvilli, or taste hairs. They're on the apical surface of these cells, and they're found in a taste pore, a small depression at the surface of the taste bud. In the membrane of gustatory microvilli are cell surface receptors that can bind to taste molecules, or channels that allow taste molecules to enter the cells. On the basal surface of these taste cells, the cells synapse with dendrites. These dendrites are going to send a signal up towards the brain. Here you can see a taste bud composed of several different types of cells, but we focus on the gustatory receptor cells. On the apical surface, they have those gustatory microvilli, or taste hairs, that come off. Again, those are the structures that actually bind to or sense taste molecules. On the basal surface of those gustatory cells, we can see synapses with dendrites of sensory neurons that will carry an electrical signal up to the brain in response to taste molecules. Now let's discuss the physiology of taste. A tastant is a chemical that stimulates gustatory receptor cells. In other words, it's chemicals that we can taste. Tastants work in two different ways. One, the molecule can actually enter directly into the cell through channels. This is how sodium and hydrogen ions work. In other words, the taste salty and sour. Notice that these ions are positively charged. If they flow into our cells, they will depolarize them. In other words, the resting cell, which is negative, will begin to become more positive. If enough of these ions rush into the cell, that will initiate an action potential. The other way that taste molecules can work is that they'll bind to cell surface receptors. Cell surface receptors are in the membranes of our taste cells. A taste molecule will bind to these receptors, and these molecules include the sweet, bitter, and umami flavors. When a receptor is bound to a taste molecule, it will activate nearby proteins, and those proteins will activate or open up ion channels. Ions like sodium are going to flow into the cell, causing it to be depolarized. Notice that this process of depolarization is indirect. The taste molecules remain outside of the cells, and different ions have to rush into the cell. Once an action potential is carried out, on the basal surface of taste cells, neurotransmitters are released. Those neurotransmitters will diffuse across the synapse and bind to the dendrites of target sensory neurons. Those sensory neurons will be stimulated and send a signal up to the brain. This diagram shows a taste bud with different gustatory receptor cells being stimulated by different tastants. Here we can see a salty or sour receptor cell. It's being stimulated possibly by sodium ions. Again, that will initiate an action potential in this taste cell. The action potential will trigger a release of synaptic vesicles. Remember, synaptic vesicles are loaded with neurotransmitters. In this case, the neurotransmitter is serotonin. Serotonin is released into the synaptic space or cleft, travels across that cleft, and binds to the dendrites of a sensory neuron. The sensory neuron is now activated and will send an action potential to our brain. Then we perceive that as salty taste. Section 2, olfaction. Olfaction is the sense of smell. An odorant is a molecule that can be smelled, just like a tastant was a molecule that could be tasted. So like taste, olfaction is a chemical sense. Olfaction occurs in the nasal cavity. This is the inside of our nose. On the upper surface of our nasal cavity is the olfactory mucosa. This area contains millions of special cells called olfactory cells. Olfactory cells can detect thousands of different odorants. They have much wider diversity than the sense of taste. Olfactory cells are actually types of neurons. In the diagram to the left, you can see a mid-sagittal section of a person's head. This large area here is the nasal cavity. So as we inhale, odorant molecules are brought upward, up to the nasal mucosa. You can see over here we've zoomed in on the nasal mucosa. You can see those olfactory receptor cells. Notice they are bipolar neurons. The dendrites dangle down into the nasal cavity, where the axons actually poke through little holes in the skull, known as the cribriform plate, up towards the olfactory bulb, which will carry signals to our brain. Now let's discuss the physiology of olfaction. I'd like you to know that smell contributes more to flavor than taste does. Remember, we could only sense 5 different tastes. We can sense thousands of different types of smell. And flavor is the overall impression that food gives us when we eat it. Flavor can include smell, taste, and also perhaps the feeling of pain, which is stimulated by spicy foods, the feeling of dryness or moistness, depending on the moisture content of food. All of these things contribute to the overall flavor of a food. Odorant molecules bind to receptors on olfactory hairs. Olfactory hairs are long projections or cilia that dangle down off of olfactory cells. You can see here in this animation. Here are the olfactory hairs dangling down into the nasal mucosa. When odorant molecules bind to these cilia, it activates something called a second messenger system, specifically the G protein system. You don't need to know the details of this system. This is something that might be discussed in a subsequent biology course. When odorant molecules bind to these cilia, it opens up sodium channels. Sodium will rush into these cells, depolarizing them, making them more positive on the inside. And this might initiate, if the signal's strong enough, an action potential. That action potential will travel down olfactory nerves up into our brain. Here you can see the cellular processes that occur during olfaction. Please don't worry about all of these different details that you can see on this slide. The main takeaway is that an odorant molecule, that is a molecule that we can smell, will bind to a receptor on the surface of an olfactory cell. That stimulates the receptor, which will activate a whole bunch of proteins that you need not know about, but those proteins will lead to the opening of a sodium channel. When the sodium channel is open, it allows sodium to rush in to the cytoplasm of the cell, causing the cell to become depolarized. If enough sodium rushes in, if the signal is strong enough, an action potential is initiated, which will eventually lead to the sensation of smell. This diagram is just meant to show you the location of these various structures. Again, here's your nasal cavity to the right, the nasal mucosa at the upper surface of the nasal cavity. We can zoom in this area here. You can see, again, the perforated portion of the skull called the cribriform plate, where the olfactory receptors poke through. The dendrites hang down into the mucosa, picking up odorant molecules, sending their signals up through the cribriform plate to a bulge in the nerve known as the olfactory bulb. The olfactory bulb contains a bunch of cell bodies and dendrites of sensory neurons. Those neurons are going to carry their information up through cranial nerve 1, or the olfactory tract, into the brain. Once olfaction is stimulated, the olfactory nerve sends information to the olfactory cortex within the brain. Specific odors come from the activation of many different receptors. What I mean is that the odor for strawberry comes from many different molecules from within that strawberry. Olfaction is tied to the limbic system in the brain. This is the part of the brain that's tied to strong emotions and long-lasting, long-term memories. So the sense of smell is tied to memories from our past. The smell of cookies might make you think of your grandmother baking. Olfaction is highly sensitive. Just a few molecules can activate the sense of smell. And that concludes part 1 of the voiceover lecture for Chapter 8 on the special senses.